Hi, this is Alex from devstories.eu. Scanpy single-cell analysis, episode 9 of 16. Traditional clustering algorithms assume your data exists in perfectly round blobs organized around a central point. But real biological data is messy, interconnected, and forms complex continuous shapes. When you try to force those complex structures into simple spheres, you end up splitting single cell types into artificial fragments. Clustering with Leiden solves this by looking at how cells connect to each other rather than just where they sit in an abstract space. People often default to thinking about clustering like k-means, where you define a center point and group everything nearby using standard physical distances. Leiden does not operate like that. It is a graph-based clustering algorithm. It completely ignores Euclidean distances to a centroid. Instead, it relies entirely on the density of edges in the neighbor graph you built earlier in your analysis. Think of the neighbor graph as a massive social network. The cells are individuals, and the edges between them are friendships. Leiden performs what is known as community detection. It searches for groups of cells that have a very high number of connections within their specific group, but very few connections to the outside network. To achieve this, the algorithm optimizes a metric called modularity. Modularity measures the density of links inside communities compared to the links you would expect if the network were totally random. The algorithm starts by assigning every single cell to its own individual community. It then iteratively merges these communities, moving nodes back and forth, constantly checking if the new grouping increases the overall modularity score. Leiden is specifically designed to refine these partitions carefully, guaranteeing that the final communities are densely connected internally and not suffering from disconnected fragments, which was a known issue in the older Louvain algorithm. In Scanpy, you run this using the tool function Leiden. You pass it your main data object and it operates directly on the existing neighbor graph. The output is a new categorical column added to your data containing a cluster number for every single cell. Here is the key insight. The most important control you have over this entire process is the resolution parameter. This parameter acts as a dial that dictates how aggressively the algorithm splits groups. By default, Scanpy uses a resolution of 1. If you increase the resolution value, you get more clusters. The algorithm becomes highly sensitive, breaking the graph into smaller, highly specific subpopulations. If you decrease the resolution value, you get fewer clusters. The algorithm becomes more tolerant, grouping larger portions of the graph together. Suppose you run Leiden and look at the resulting map. You might notice that a single known biological cell type has been split into 5 tiny overfragmented subpopulations. The algorithm found slight differences, but biologically, those 5 groups belong together as one distinct cell state. To fix this, you simply run the Leiden function again, but this time you explicitly pass a lower number to the resolution argument. By dropping the resolution, you instruct the algorithm to relax its criteria. Those 5 tiny fragments will merge back into one solid biologically meaningful cluster. The exact resolution you need is never a fixed mathematical truth. It is an adjustable dial you turn until the statistical communities in the graph accurately reflect the biological realities of your tissue. Thanks for hanging out. Hope you picked up something new.